very much for inviting me as the final speaker, keynote speaker. It's a great, certainly a great honor, and has been very nostalgic to be back here with all the colleagues and the amazing science that's going on in Dresden. So, yeah, I just thought I'd talk about understanding the signaling architecture underlying regenerative patterning systems. And actually, I'm going to mainly talk about unpublished work. In 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 our lab, we've become very interested in understand. Oops, I can't really see anything here. The self-organization and regeneration of organizing centers from, let's say, random or variable starting points. And regeneration is super interesting in that way, in that, for example, in nature, an axolotl is bitten by its neighbor, and you can have an extremely jagged amputation plane, yet you form a blastema. That blastema undergoes patterning, and it regenerates a beautiful limb. And we all know that by now that regeneration is a recapitulation of development in which you have to then start expressing morphogens in defined locations in order to get this patterning. So we've been interested in this problem, both looking in the spinal cord and in the limb. And I'll mostly talk about, oh, I hope I can get this right, the limb, but I just wanted to point out a little bit of the work that we have done and are doing looking in the spinal cord. And so in development, we know that the floor plate is an extremely important organizing center. It's a group of cells forming in the neural tube that expresses FOXA2 transcription factor and sonic hedgehog that is secreted to the neighboring cells in order to generate an array of 11 different progenitor cell types along this neural tube. Of course, there's a counteracting signaling center here in the roof plate that contributes to the array signaling that generates this array of cells. And in development, which is very regular, of course, it's canonically understood that the notochord is secreting itself sonic hedgehog that induces this floor plate to form in this place. Now, in regeneration of the spinal cord in the axolotl, you grow a new tube of neural progenitor cells from the amputation plane. And this is a cross-section of a regenerating axolotl spinal cord with the floor plate. And you can see that there's sonic hedgehog being expressed here in white, but there's no notochord underneath. And so we became interested in how you may be able to form a floor plate without the classical inducing center. And we've done some work on the axolotl spinal cord, but actually that got us interested in asking whether we could reproduce such non-notochord mediated floor plate generation from mammalian neural stem cells. And so for a while, Andrea Meinhardt was trying to generate neural stem cell cultures. In this environment of epithelial biology, we were trying to make basically neural cysts that make this three-dimensional structure from mammalian neural stem cells. But she never could do that. And early on then, she realized she had to go forward in this process. So she started with single mouse embryonic stem cells, put them in matrigel in order to make three-dimensional cysts. And then she went forward, and indeed she got beautiful, what we called neural cysts. And if she added retinoic acid at day 2 in order to posteriorize the organoids, she magically got these neural organoids that had a floor plate in one part of the organoid. And this is a functional floor plate in which you can see here the expression of sonic hedgehog and the generation of progenitor domains around this sonic hedgehog floor plate. And so Teresa Kramer and Hannah Stewart, who are really exceptional student and postdoc, have been working on this system to understand how a global pulse of retinoic acid induces this localized floor plate formation. And I'm not going to talk that much about it today. We just put this out on bioRxiv. They somehow misspelled my name in the preprint, but yeah, this is it. This is just one example. I hope I can. This is just one example of the type of data that we've collected to understand the self-organization process, where we have a FOXA2 venous fusion protein reporter, and we film the process of this floor plate organization. And we see, as Pierre Francois described, both clustering of cells, but also this amazing competition between protoclusters that we see during the process. And so I hope this should run. Oh, no. So you see 3 clusters forming, and then 2 of them disappear, and then 1 survives. And so by statistical analysis, we could infer that the clusters were competing with each other, and by doing chemical screens, we could identify that BMP is the main molecule that is mediating this cluster competition. And you can read more about that here in this floor plate, in this preprint. Now, getting back to the main subject of our lab, the axolotl, it's always still amazing to see how this axolotl can regenerate. So here you have an intact limb, you amputate it, and then over time you see the limb regenerating and reproducing its pattern. And in an amazing aspect that of this regenerative system was revealed by transplanting a blastema from the left side to the right side. And in this case, then you get 3 limbs regenerating. And this was interpreted as meaning that you have positional information in the anterior and the posterior side that's stable, so that when you transplant this blastema from the left to the right, you're generating 2 additional interfaces between anterior and posterior tissue. And this is a signal that a new limb should grow out here. And so it's interesting to think that this actually is probably a way in which you gain amputation specificity, so that you won't regenerate a limb if you just make a wound in the side of the limb, because we all know that wounding the tissue is an extremely important initiator for regeneration. Now, we were really fascinated by these kind of grafting experiments, and over time, then Eugene Naku in the lab could show that this anterior-posterior requirement is represented by the fact that when you amputate the limb, posterior cells are poised and turn on sonic hedgehog, and anterior cells are poised to express FGF8 when they get into the blastema. And as described in the studies of the developing vertebrate limb, then sonic and FGF form a positive feedback loop to each other, so that the FGF8 expression is dependent on sonic hedgehog, and sonic hedgehog expression becomes dependent on FGF8. And an interesting aspect of this is that all vertebrate limb buds express FGF8 here in this ectoderm, but it's the salamanders express this FGF8 in the anterior mesenchyme. And so we think that this is an adaptation of this system, this limb development system, that on one hand provides amputation specificity to resuscitating this developmental system in the adult after amputation. And also we think that this might be helping in scaling the whole system. So as you'll see, axolotls of many different sizes regenerate. And if you have just a stripe of FGF8 expression here in the ectoderm, we may expect that at some point you'll reach a size limit in the ability for this system to work. Whereas if now you're expressing FGF8 in a volume of mesenchymal cells, that this may now, the system may now scale better. So Leo Itsuki's postdoc in the lab, who's been interested in understanding the memory aspect of this anterior-posterior system. So he's generated a lot of fancy transgenics to follow the cells that had expressed sonic hedgehog, for example, during development. So this is developing limb bud. And so he has a direct reporter plus a Cre-ER in a double transgenic with the LOXP reporter. So when he injects the animals with, or treats the animals with tamoxifen during development, he can mark the cells that are expressing sonic hedgehog, and you can see the direct expression in blue here. So you have blue and red during development. So then when he traces these cells out through the formation of the limb, you see this beautiful stripe of cells that had expressed sonic hedgehog during development, and you see always this full formation of labeling of the last digit and half of the second to last digit. Interestingly, when people have done this experiment in mice, you only get labeling of the hand area. And so it's interesting for us that in the axolotl you see it throughout the limb. Now, Leo's done a number of experiments to ask whether when you amputate the limb, do all of the new sonic hedgehog cells come from the cells that had previously expressed sonic hedgehog? And basically the answer is no. You can actually see it without doing any manipulations. He's actually removed the sonic cells and shown that you get new sonic cells in the regenerate. But you can see here after amputating a blastema, you're getting blue cells that are not red. And if you look in an even larger blastema, you see here blue cells that are not red. And so that means that there's a larger field of cells that are poised and competent to turn on sonic hedgehog that had not turned it on during development, that then migrate into the blastema and are able to turn on sonic hedgehog. And this is something that Leo is working on. He's identified a transcription factor that seems to mark these a HAN2 that marks these cells in the mature tissue and that is necessary for the onset of sonic hedgehog in the regenerate. And so we're interested in how the onset of HAN2 expression is controlled by injury. And of course, a very important question is how is the size of this zone set? So you can see it's very small in a small limb, and it's large in a large limb. And that's what I want to spend the rest of the time talking about today. So this is the size aspect that you can see here. So animal this size has a limb bud, and then it quickly forms a limb, so that you have animals that are ranging in tenfold difference in size that have a limb, and when you amputate it, are able to regenerate, form a blastema, turn on sonic hedgehog, and regenerate the limb. So the question is, how does it happen? So this is the work of Pietro Tardivo, a really outstanding graduate student. Oops. So what he first asked is, what is the relationship between animal size, limb size, and blastema size? So he just simply measured the length from the snout to the tail, and then he measured the width of the arm, since we're interested in the anterior-posterior specification. And then he measured also the width of the blastema at the mid-bud stage, just at the kind of junction between the mature and the regenerating zone. So you can see here a 2 centimeter limb, 11 centimeter animal's limb, and a 20 centimeter animal's limb. So you can see obviously that the bigger animal has a wider limb and that the blastema is bigger in size. But interestingly, at the 20 meter animal size, the blastema is bigger, but it's not proportionally bigger proportionally bigger. So these are the numbers that you can see that he generated. So you can see the width of the arm is scaling with the body size. And then at small animal sizes, the blastema width is also scaling, but then eventually it's not scaling with body size. And so you might be wondering then, how does a big animal regenerate its limb? And so this is just a time series of a blastema that's forming. You see that it's thinner than the size of the arm, but then you see a kind of mini limb emerging. And it's been known for a long time, and Kate McCusker has been studying this, this phenomena, which is that you make a blastema and it makes a mini limb, and then over long periods of time, this mini limb fills out to match the size of the animal. So that means that in some sense, this kind of scaling aspect it really happens in two phases. One is the size of the blastema and the size of these kind of developmental signaling events that are happening, and then a later phase where then the limb matches the size of the animal. And Kate has shown that this is dependent on the number of nerves and the type of nerves that are innervating the limb in a large animal compared to a small animal in this late phase. But Pietro was interested in this blastema. Yeah. And one other aspect that's interesting for us is that if you look at the number of animals that have a patterning defect in the digits, you have an increased number of animals showing patterning defects, so missing digits, as animals grow large. So an interesting question is whether the missing digits are related to the fact that this blastema is no longer scaling with animal size. So what determines digit number? So there's been a lot of theories about how digits form. One recent work from James Sharp's lab suggests that you might have a Turing type mechanism that creates a field of periodic elements that represent the forming digits that will then grow out. And indeed, if you remove Sonic Hedgehog in a mouse limb, you are missing digits. And then if you overactivate Sonic Hedgehog by knocking out GLE3, the GLE3 repressor, you generate a bigger field of the limb bud, and you generate more digits, corresponding with this idea that the size of the field in the end, with a certain periodicity of digit progenitor cells, will then determine the digit number. So, one possibility is that if, as animal size grows, and then you're trying to scale the blastema, but then if the size of the blastema is somehow not scaling in the same way as the digit wavelength, then you may come to errors in digit number. And this is something that we're very interested in testing. So coming back to this aspect that the blastema size is not scaling with limb or body size, a question is whether the range of Sonic Hedgehog signaling is becoming too short in the large animals, and so whether the size of the width of this blastema is basically limited by the range of Sonic Hedgehog signaling or the duration of Sonic Hedgehog signaling. And this is just to show that in axolotls, if you inhibit Sonic Hedgehog during regeneration, you inhibit digit formation, and if you overexpress Sonic Hedgehog, indeed, you get more digits. Oops. So Pietro's then interested in revisiting this Sonic Hedgehog FGF signaling loop and asking how its properties are changing with changes in animal size. So in essence, he's asking, in a large animal, which I showed you, is a sonic zone scaling with animal size? And then what about the signaling range of sonic? And this we're reading out by the expression of patched 1, which is a target of Sonic Hedgehog signaling. So is the range of patched signaling with animal size? And so he's done this by using fluorescent HCR hybridization chain reaction in situ hybridization, which is highly quantitative, and then he optimized the C3D clearing method, which really took a lot of time to find the right clearing method that would allow him to confidently be able to quantify these signals in animals of very, very different sizes. And then he's using a light sheet in order to image these samples. So you'll see here the different channels here. Here's nuclei, merged, sonic, patched, and FGF. He also images autofluorescence to be able to see the blood cells and then remove them out. So I hope it should go. Oh, I see. Oh, wait a minute. It should. Okay. So you see here, for example, like the patched 1 RNA. Here's sonic. It's in a smaller zone. And so this is just playing through the stack of these blastemas. So then you can see the autofluorescence here. These are blood cells. But you can see in white the Sonic Hedgehog mRNA. And so he performs automated image identification to identify the sonic domain and the patch domain. He extracts the volumes, and he takes basically the line perpendicular to the longest axis of the sonic hedgehog. He then generates basically a rectangle, and then it's in 3 dimensions, and collapses that information into 1 dimension. And by that, he can extract the patched signal as well as the sonic hedgehog signal in order to identify the intensity along this AP axis. And so you can see an example of this kind of information here. Now, so we've been through this with several people talking about gradient formation, gradient signaling in this meeting, as also as during regeneration. Of course, we're interested in the production, the diffusion, and the degradation of the signal. And we, you know, what type of profile do we expect? You know, when it's just diffusion and degradation involved, we may expect a linear gradient. We were also considering that we may see a power law gradient when thinking about sonic hedgehog signaling, because sonic hedgehog has this architecture where patched is actually a ligand, a receptor for sonic hedgehog that's inhibiting smoothend, and then when sonic binds, then this inhibition is released, smoothend acts, and then you induce patched. So there's this feedback where you induce more patched, you generate more negative receptors on the cell surface. And so, so when you activate with signal, you increase patch levels, and then more sonic is required to activate the pathway, and then more sonic is degraded by endocytosis. But when we looked at our data, we get a very beautiful exponential shape in the patched RNA over the AP axis. And so we can define a gradient amplitude as well as the gradient length scale in this data. And then we always define the source of sonic when it comes down. So you can see then here the data for looking whether the source of the sonic hedgehog expression domain is scaling, and you can see that there's scaling of the sonic hedgehog volume domain volume along with animal length, and he's taken this measurement out to beyond 10 centimeters. Now, interestingly, when he measures the patch 1 length scale, he sees scaling up to about 8 centimeters, but then the scaling seems to break down when he gets to animals above 8 or 10 centimeters. And so indeed, it seems that there's a limit to scaling of the sonic hedgehog signaling in these animals. So in the size ranges where he does seem to see scaling, he compares, for example, 2 centimeter and a 4 centimeter size animal. If he just, you know, normalizes that data, then he can see a very good correspondence of the data from the 2 and the 4 centimeter normalized data. And what's interesting for us is that it seems that comparing in these stages where the signaling is scaling, when he compares the data, he seems to see that the amplitude of the patched mRNA is similar between small and large animals, and as well as the sonic hedgehog. So, of course, he needed to know whether this situation is changing over time. How do we compare which time point after regeneration between the animals? And so he's looked at 3 consecutive days in regeneration. And you can see that it is changing, that the pattern of sonic hedgehog, these are different animals, of course, but on average, the pattern of sonic hedgehog is changing over time. As, for example, you start differentiating the bones, then eventually the system shuts down, and then the animals continue to undergo differentiation. And at the moment, we're still collecting this data, but it looks like the sonic hedgehog expression and the patched expression over time are either staying the same or may actually even be increasing over the 3 consecutive days in regeneration. And this is the amplitude. But interestingly, if he looks at the length scale of the patched in these consecutive days, it's either staying the same or going down. And so we're not having a situation where over time this gradient is kind of getting bigger and bigger in these animals. So you can see the data here, the patch gradient length scale, the patch gradient amplitude, and the correspondence of this data. So we, of course, have to take many more examples to be really sure about this data. But one thing that we're sure about is that the gradient is not getting longer over time in regeneration, in the time course of regeneration. So in order to investigate that further, Pietro expressed more sonic in the animal. So he used a baculovirus to target sonic hedgehog expression in the sonic domain in order to just boost the amount of sonic that's being produced. And there, when he does that, if you compare the normal with the boosted sonic hedgehog, then indeed he can see a shorter length scale when he has increased sonic hedgehog expression. So that means to us that you cannot really explain a larger length scale in larger animals by the fact that you have a bigger or more sonic hedgehog production. So what I'd like to point out is that, this is an amazing system. We think that there's perfect scaling of the patch gradient within some size regime. That means that the amplitude of the patched and the sonic are not changing over different size animals, but we're getting a scaling of the gradient along the AP axis. We see that blastemas from larger animals have larger sonic hedgehog domains, and that the length scale of the patch gradient scales linearly with animal size in animals that are smaller than 10 centimeters, but appears not to increase in larger animals. So that means the blastema size and the blastema patch gradient from large animals are underscaled. And of course, the obvious question is this related to the defects in digit number? So as I mentioned before, then we just want to point out that a larger sonic domain is not sufficient to increase the gradient length scale. We showed that when we increase sonic, you actually reduce the gradient length scale. And does the size of the blastema determine the gradient length scale? No, over time in regeneration, the blastema size seemed to increase, but the gradient length scale does not. So we've been thinking about this problem with Ben Friedrich and Natalia, his student, and they've been thinking about different ways to scale the sonic hedgehog gradient. And so then, of course, we have to take into account the diffusion and the degradation rate. And so they've been thinking about the properties of dynamic scaling with tissue size, meaning with growth over time, versus static scaling, where the scaling has to do with animal size. And so they started a lot of this work before we had all these results. So this is rapidly changing as we get these results. But in essence, what they've been interested in is, how does this field of cells along the AP axis then grow in response to the presence of these morphogens? So they thought about a growth rule where you need 2 morphogens, the sonic and the FGF, and then that both morphogens have to remain over a certain threshold in order to sustain growth. So then as the field grows, then you may be eventually coming down to a situation where the 2 morphogens are now below the threshold, and this is then how growth may stop. And as I mentioned, these 2 morphogens are cross stimulating each other. And they considered, in thinking about how the system may work, whether it would work with a single morphogen, or whether you do need actually both morphogens in order to get proportional growth arrest, where the final tissue size is proportional to the initial size. And so, in essence, what they're doing is a model screen, minimal model screen, and asking if they think about scaling with either the diffusion coefficient, the degradation term, or mixed scaling, which one of those scenarios theoretically then matches with the experimental data. And so when they're thinking about scaling with growth, really they cannot find let's say, a condition that fulfills the fact that what we see is that the amplitude is not changing with animal size, but then to get proportional growth. And so these are the different scenarios, and it's actually quite difficult to get proportional growth when you're thinking only about scaling with as the animal's growing. But then in terms of a scaling factor. But then if you think about a scaling factor that would actually operate depending on animal size, then they're able to find conditions in which you can get scaling of the gradient with animal size, but for example, fulfill the condition that we see that the amplitude of the patch is not changing with animal size, and that you're getting proportional growth proportional growth of the blastema with respect to animal size. And in screening all these conditions, what they seem to find is that the way it would work would be if you have a scaling factor that affects diffusion, and that you have two morphogen component system in order to get this proportional growth. And so that makes a strong prediction that, that we would have a scaling factor that affects the diffusion of the sonic hedgehog, and that is dependent on animal size, and that's not a kind of feedback factor that would be then changing as the animal grows. And so that's something that we're now working on and searching. So before actually they came up with all of these different scenarios, we have started looking for scaling factors. So for example, classic possibility would be SCOOP2. It's a secreted protein that interacts with the cholesterol moiety of sonic hedgehog. It facilitates the release of sonics, so you might think that it's very good at getting sonic to pass through the extracellular space. Another possibility that we had considered was GAS1, which is a co-receptor. In some cases, it's also thought to be the shed from the plasma membrane, and then sensitizes cells for sonic hedgehog signaling. So we've been making mutants for these kind of factors, and you can see what we do is we inject a gRNA into the egg of the axolotl, and then in the F0 generation, you get mosaic animals. And so you can see an animal like this here, where the left limb looks pretty normal, but the right limb is quite shriveled and small. And so we're growing these up, and we're getting F1 animals, but these are different examples of these kind of shrunken limbs. Here's the data for GAS1. Again, here's the control, but then here you can see small limbs in the developed animals. And we get a loss of anterior structures and a certain number of crispin animals. But I must say that so far in these mutants, for example, you can see here when we look at patched 1, here you can see that GAS1 is actually inversely, the expression pattern inverse to the patch 1, so now we would say it's unlikely to be such a scaling factor. But although we can remove GAS1 from the limb bud by this gRNA expression, the patched 1 length scale has not changed at all. So that means GAS1 is obviously playing a quite different role. We have some ideas what it's doing in this whole situation. So at the moment, we're asking, for example, whether cells from large animals and small animals have different sensitivities to responding to sonic hedgehog. And of course, through these kind of miraculous transplantation experiments, we can transplant cells from large animals into small animals and vice versa to ask whether the features of responding to the sonic hedgehog gradient are autonomous or non-autonomous. And so this is ongoing work. So I just want to stop here. So we're very excited to have generated this data of this remarkable scaling. Even though it breaks down eventually, it's still a pretty remarkable scaling system that depends on animal size. So animals, blastemas from large animals, you do get scaling, but eventually at large animals, it's underscaled. You get defects in regeneration more frequently in these larger animals. These blastemas from larger animals do have larger sonic hedgehog domains, but the length scale of the patch gradient scales linearly with animal size up to 10 centimeters, but then it all seems to break down. And so we're very interested in understanding whether the scaling of gradient length scale determines the scaling of limb regeneration and whether it's related to the failure of scaling in the number of digits that these animals make. So I want to thank these amazing people who have been in my lab, who are in my lab doing these experiments. It's also been a great collaboration with Tatiana and Ben and Dunya and Natalia. And I also wanted to mention that we're working with Fabrizio and Eduard on other aspects, on aspects of patterning and length scale in the limb regeneration also. And thank you very much. Yeah. Yeah. Yeah, so based on our HCR measurements, it seems that the signal per cell is very similar between large and small animals, which was a great surprise, actually. Yes, yeah.
Very nice, Sally. So I was wondering whether the very big animals, are they not reproducing anymore? Are they bitten less? Or is there any adaptive reason for which, very big animals could be less able to regenerate as well? Or it doesn't really matter if you have more or less digits, it's because
Yeah,
you'll still
yeah,
cope with life.
right. Yeah, I mean, they probably don't rely on their digits all that much. And, I think that the salamanders regenerate well and also because they have a tail. And so if you don't have a limb, you're actually still fairly functional because you can swim around and get food. And so maybe, at some point, having the perfect number of digits is actually not that, necessary. Yeah, yeah.
But are those big animals common in the wild?
Yeah, there are.
Yeah.
So when people look in the wild, they do see a number of animals with missing digits. Yeah, digit deformations. Yeah.
Yeah. Yeah. So I was really impressed by your talk, and I was curious about the transplantation experiment. Considering that the gradients are still set up,
Yeah.
what is the chirality of the extra limbs you're generating, and why do you think you're generating a third limb specifically?
Oh, so you mean in terms of this transplantation experiment? Yeah. So actually, the orientation, the AP orientation of the limb is corresponding to what's shown in that diagram. So in essence, okay, so on the anterior surface of the mature, now you're bringing posterior cells there, and so in essence, that posterior tissue is signaling to anterior tissue in the stump, and then generating one limb, one hand is the same, and the other hand is the other direction. Yeah, yeah.
So I just had a quick question. I think maybe you said it, I might have missed it. What's the mechanism that actually sets basically the number of sonic hedgehog secreting cells in the first place? So how do we know exactly
Yeah.
how many they actually
Yeah, no.
given the size? Is it just basically some fraction of the cells, and then you basically, if you have a set fraction, you
Right.
always generate the same amount of sonic hedgehogs? Or do you know
Yeah.
that mechanism?
No, we do not. I mean, in limb development, it's known that you need the cells need to be near the epidermis in order to turn on sonic hedgehog. And indeed, in our case, at least at the beginning, the new sonic the zone of sonic is underneath the epidermis. As said, you know, you're obviously getting cells that are not at the most posterior side, kind of migrating up to the posterior side, and they're turning on sonic. So we think that there's a communication with the posterior epidermis, but we don't know very much about it. I think that's a super interesting and important
Maybe it's
question.
somehow related to this. Okay.
Yeah.
And also, just if you could speculate really quickly, this is a very naive question.
Yeah.
Do you know why don't humans do this? So,
Yeah.
yeah. So
Yeah.
what's
Yes.
the
Yes.
modeling?
Yeah. Actually, so we work on those aspects. So what we can, so the axolotl does this because the connective tissue fibroblasts de-differentiate into limb bud progenitor cells that becomes multipotent with respect to connective tissue lineages. So then, and turns on the whole developmental program again. So what we've been seeing, we've been working on the mouse, and if we do heterochronic transplantations of mouse fibroblasts into the mouse limb bud, the mouse fibroblasts are unable to de-differentiate into this fully potent cell type. What's interesting is as I mentioned, that Leo has been looking at signatures. He has a signature of this pre-posterior cell in the mature state, and so he can see a series of developmental transcription factors that are there, kind of poised to turn on. And when he looks in mammalian mouse fibroblasts, it's interesting. Those factors are expressed in adult fibroblasts, but they seem the whole set of them seems to be distributed to different subpopulations of fibroblasts, whereas in axolotl they seem to be all in the same fibroblasts. Yeah, so there are a number of things that could be preventing the mammalians from doing that.
Hi, thank you for the very interesting talk. I have two quick questions. So first one, you showed the expression pattern of GAS1, but I don't think you showed the expression pattern of SCOOP2.
Yeah.
Right. So what is SCOOP2? Where
Yeah.
is it expressed, and does it, how does it work in
Yes,
terms of
yeah.
relative?
So yeah, SCOOP2 is actually quite uniformly expressed in the limb. We don't yet know whether the amount of SCOOP2 is changing with animal size, but in that sense, SCOOP2 would be a better candidate, could be a better candidate for a scaling factor. But as I say, we have not yet detected that the patch length scale is changing by altering SCOOP.
I see. And if I may ask a second question, if I remember correctly, in the beginning you seem to say Sonic Hedgehog and FGF8 seems to have a positive feedback.
Yes.
And they reinforce each other.
Yeah.
So how does that positive feedback play in a role when the tissues start growing larger and separate? Like, how do you?
Yeah, so this is why, sorry, I didn't explain it very well. So that's the idea, right, is that if over time the tissue's growing, the blastema's growing, then these signaling centers are getting further and further apart. And we think this is one reason or one way that the tissue stops growing and you get proportional kind of, proportional. The idea is, how do you then get proportional stopping at the right size with respect to the initial conditions, right?
Thank you.
Yes.
But it was still a very linear curve.
Yes. Yes. Yes.
So you could still a type of scaling, but with an offset. So
Yes. 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 We don't. We don't really know. There could be other factors involved in the scaling. For example, the number of nerves may also be contributing to the number of cells that go in and that contribute to the size of the blastema. Yeah.
do we have further questions? There's one here.
Let's give a very naive question. The developmental error you get is that if you have a too large animal,
Yeah.
too large blastema, you seem to get missing limbs
Yes.
rather than too many digits. So do you have an idea why it's in that direction, not the opposite?
Yeah, as mentioned, I mean, I guess we assume then that we don't know yet how. I mean, when the digits start forming, you get condensations of cartilage cells, and we don't know yet whether the size of these cartilage condensations are increasing with animal size. But we expect that maybe this aspect is scaling differently than the total size of the blastema.
So maybe you get the errors because some aspect is compensating much more or a bit too much compared to the other.
Yeah, I don't know whether it's
manipulated.
compensation, but that there might be a mismatch in the way that different aspects like differentiation versus the blastema size control are scaling with animal size. Yeah, we can talk about it some more. Yeah.
Pierre has a last question. I might have a shout then. When you were showing the size of the blastema against animal size,
Yeah.
it looked as though the variability also increased with animal size.
Yes.
In the data, and is that also reinforcing this notion that some of the animal cares less about having the right
Oh.
blastema than having the right number of digits, for example, as it gets larger?
Yeah. Yes, yeah, it probably is more I have to admit, I can think about ideas, but we haven't thought about it that much actually, in terms of the variance of the blastema size, and whether whether that might be an human experimentally induced situation or whether it's a true aspect of the system. I think that's something we have to be very careful about. Actually, we're even still a little bit, although I discussed, the breakdown and scaling of the blastema, we're still a little bit hesitant that there could be aspects of this situation that are human experimentally induced. So I do think it's something we should be a little bit careful about. Yeah.